So now you can just move on.
All right. So thank you so much, Heiko, for that introduction. So as Heiko said, I'm going to talk about a computational model to do with emotion. And to some extent, when you think of emotion, you don't immediately think of mathematics or computation. So it can seem like an unusual approach. But what I'd like to do is take a very big, broad perspective and talk about very broad ideas about what computational modeling is and the different types, and then kind of work my way towards this emotional gatekeeper or EmGate model. So if there's any clarification points, you can maybe feel free to interrupt me or ask a question via Heiko in case I don't hear it. So with that, we'll get started. So my plan is to start very broad and ask what exactly is a model. Sometimes I'm sure many of you have seen GIFs like this. When you think of computational model, you see scary mathematical symbols, especially if you don't come up from a physics or computer science or math background or engineering. And it seems very different from a wet lab or something in the psychology lab. And it can be a little disconcerting, but I'd like to sort of say that it's not as complicated as one might think, even if you don't have that much of a math background. Perhaps more complicated is the question of what is an emotion. So I'll offer some tentative answers because it can be a topic with a lot of debate. So from there, I'll make some comments about how to go about building a model from various pieces, experimental data, theoretical ideas. How does a model come out of all this? And in conjunction with that, I will talk about how to use a model, and specifically this emotional gatekeeper or EmGate model that my colleagues and I developed. So we'll use that as an example. So there's a kind of bird's eye perspective, and we'll work our way towards more detail. There won't be any equations or math. It'll all be at the level of concepts. So what exactly is a model? Sometimes when I was in grad school, we had people who joined and wanted to know what is a model, how do we go about it, and they wished there was a textbook. And some of the older students were very confused. But in retrospect, I think there are things that you can prepare a student with because it can be disconcerting, even if you are fairly good at the mathematics and coding. So models in some sense precede science. We have, for example, evidence of these. This is a model of a granary from ancient Egypt. We don't know whether it was used by architects or purely for ritual purposes. But then later we have things like this, which are models of celestial events. Or we have something like this, which is an orrery. It's a mechanical model of the movement of planets. And things like this have been around for a very long time, used by architects and eventually by mathematicians and astronomers, even before what we call the scientific revolution. So when you think about a computational or mathematical model, it's always good to sort of remember that ultimately it's like making something that resembles something else, whether it's with physical objects or computational processes. So among mathematical models, there's a kind of spectrum. On the one hand, in biology and psychology, you'll often come across statistical models. These are models used to summarize the data, but they don't necessarily make causal assumptions. I'm simplifying a little bit here because it's getting more complicated lately. And then at the other extreme, you have dynamical models of the sort that I'll be talking about. So these models attempt to show how the data might arise in time, and they can be used to summarize what we already know of causal interactions and explore further implications of what we already know. And it's not a binary categorization. There's a kind of continuum between the two. And once again, we can think of these with mechanical analogs. So some of you may recognize this. This is a Galton board. Francis Galton was interested in demonstrating how a normal distribution would arise. So he used little ball bearings, which would, because of their slight differences, end up forming a Gaussian or bell-shaped distribution. And so you can show this mechanically. This is a mechanical generator, if you like, of a normal distribution. And the key idea here, when you're making a model of this sort, is that the data are assumed to involve random variables. Somewhere in there, you're drawing numbers from a hat in some sense. And at the other extreme, just like the orrery, these mechanical deterministic sorts of models initially, the idea here is that when we do them with math, they're governed by either differential equations or difference equations. And if you like, you can add randomness if you know that there's a lot of noise in the system, but it's not obligatory. Both these can be used to fit data. So you're collecting some measurements and you can use either of these, but how you go about interpreting what you've done with the fit can be quite different. But like I said, it's a continuum, and you can have models that are sort of a blend of the two, or with a focus on one or the other of these two sides. Another way to think about this, which overlaps somewhat with dynamic versus statistical, is forward versus reverse modeling. So there's a nice paper here by Jeremy Gunawardena that talks about this in biology. It's interesting and kind of slightly funny paper also. He calls models in biology accurate descriptions of our pathetic thinking. But anyway, his idea is that we can distinguish two kinds of modeling, forward and reverse. Reverse modeling starts with experimental data, some measurements, and seeks potential causalities suggested by the correlations. So as you can see, it's closely related to statistics. It's very data first. You work from data to a phenomenon. And then you have forward modeling, where you start with some known or suspected causalities, and then you try to make predictions. So you extrapolate from those. And outside of biology, we can come up with a couple of examples here. So consider astronomy. A reverse model might start with some astronomical data collected over the centuries. And from there, you could work backwards or towards the thalamic model, for instance, which is really a way of just capturing what's observed and not much else. And there wasn't really an interpretation in terms of causality, or that was sort of optional. Going in the forward direction, you have Newton's laws, so you start with Newton's laws, and you can work towards not just fitting the available data, but for instance, predicting something. So a very famous example of successful prediction is the discovery of Neptune. It was described as being discovered with the point of a pen. So there was an anomaly in the observations of the planets, and there was a physicist who used Newton's laws to accurately infer there must be a planet there. And that's a classic example of forward thinking. So we see this kind of thing in biology and neuroscience too. When we talk about models, it's really important to remember this line, sometimes attributed to a statistician, George Box. All models are wrong, but some are useful. So, and when this comes up, one often sees this sort of miniature short story by Jorge Luis Borges. He talks about a kingdom where they are exquisite map makers. And in that time, their maps became more and more complicated. And eventually, they corresponded with the size of the empire itself, point for point. The following generations, who were not so fond of the study of cartography, saw this vast map was useless and not without some pitilessness. The reason for this was that a map that is as complicated as the territory is of no use. What we want are models that are simpler than reality and therefore useful. So you could, instead of waiting around for things to happen, we can get a prediction in advance, or we get some sort of conceptual generalization. Those are all forms of simplification. Now, it's important. I'm going to be showing you a fair number of diagrams, and they're not always going to be consistent. This is something you often find in neuroscience, that even two diagrams of the same circuit will have slightly different regions in them. So you have to remember that maps, diagrams, analogies, metaphors, they're all forms of model. And there's a great line from Arturo Rosenbluth, who was an early cybernetician and colleague of Norbert Wiener. He said, the price of metaphor is eternal vigilance. Because whenever you simplify something, you're going to leave something out, or exaggerate something, or downplay something that may well be in your data already. So with that caveat in place, let's move on to modeling emotion. It's not something we often think about modeling even now. So let's start by asking what exactly emotion is. We can sort of discern two broad, again, a sort of spectrum. Charles Darwin kind of initiated a modern and naturalistic study of emotion in this famous book, The Expression of the Emotion in Man and Animals. And he was stressing the universality of many of the emotions common to man, humans, and animals. And then you have something like Lisa Feldman Barrett's work from the past few years, which is very much stressing how culturally emotions can be quite different from each other. And we're not necessarily all that good at recognizing an emotion in a very different culture. So there's these debates and back and forth, say between, for instance, the issue of invisible feelings that you don't necessarily see versus these outward expressions or behavior. And as it turns out, we're not always very good at identifying the inner feeling using just what we are able to observe. Then, as I said, there's this dichotomy between universal emotions across humans and animals, as well as among humans. How much are they culturally constructed? And there's interesting evidence for both sides. Lisa Feldman Barrett talks a lot about culturally specific manifestations. And there's also these debates about whether emotion is just a passive perception of some bodily state versus some kind of active cognitive construal or an interpretation of what's happening to you. So in my experience, it's always good to say that in these kinds of debates, there's an element of truth in both sides, and the creativity requires finding out how to combine them. So from the perspective of experimental science, we really have to focus on external observable manifest behavior expression. And particularly in animals, we can't even ask them how they feel. So there are no questionnaires. So there are some broad things that the community has settled on, emotional actions and reactions, the bodily changes that are easily observable. In the case of humans, we have smiling, frowning, blushing, eye movements, and other sorts of body language. For different species, we've come up with equivalents. In some cases, for instance, smiling, it's not clear that there are equivalents to smiling in any other animals, actually. And then there are internal changes that we don't typically see, but which we can measure, such as changes in heart rate, so different forms of arousal, so blood pressure. And then now we can talk about chemical changes, rushes of adrenaline, stress hormones. These are things, all of which, of course, are ultimately correlated with subjective experience to some extent. And then there's emotional learning and perception. So here we study the ways that signals from the body or from the external world in conjunction with some appraisal or inference come to trigger an emotional action and a reaction. And another important field, which I won't really get into, is studying how people recognize emotions in other people. So we'll focus on a couple of these simplified versions of this. So now, even if you have some of these behaviors and you've accepted that a particular sort of movement or reaction counts as anger or fear or happiness or satisfaction, how do you go from those behavior level categories to neural phenomena? How do we link it with the brain? The general goal is to figure out which parts of the brain correlate with these phenomena. And also in the case of humans, we can look at what treatments happen to work. Very often treatments are discovered before we have any theory for what's happening. For instance, in the case of Parkinson's disease, L-DOPA was discovered to help long before we had any picture of what exactly dopamine was doing. And of course, we look at how emotion-related behaviors are mediated. It says my internet connection is unstable, so sorry about that. Now this word mediated, we often sort of start by taking for granted certain things that have come from the literature. Once, some years ago, I was answering questions on Quora's website for question and answers, and someone asked a very good question. in neuroscience, how accurate is it to say that a brain area or circuit mediates, evaluates, or is the seat of a particular facet of human experience? This is a tough question. And to this day, well-respected neuroscientists have sort of pitched battles about whether functions are very, very localized or whether they're very distributed. And I don't have a strong opinion about this. I think that some functions are localized and some aren't. But the key idea is that our different methodologies are sort of like different lights shed on the same object. So this is the cover of a famous book, Gödel'scher Bach. And what I like about it, for the purpose of this analogy, is that you have three different kinds of shadow, the G, the E, and the B, which you can see depending on where you project light from. And the idea here is that different methodologies studying the same phenomenon are seeing different shadows of a complex object that we have to infer. So it's like trying to find out what the shape of the block is, whereas all of our data are just these shadows. And the big challenge of theoretical neuroscience and computational neuroscience is to come up with some object that makes sense and that produces all these different observations. So that's just another sort of caveat about saying when someone says that a particular brain area mediates something, it's really a projection. And it could be that as we get further understanding, we'll have to modify. So all these are sort of tentative in many cases. So getting into more detail, how did we start learning about localization of brain function? Unfortunately, a big part of this started with injury. For instance, with the Civil War, bullet wounds to the head revealed that brain injuries could strongly affect emotion, cognition, and personality, as well as other functions. So in this paper, they talk about how this in the middle of the 1860s or so, increased knowledge about cerebral localization was developed in conjunction with the ability to treat people. So once upon a time, the wounds were very large and people rarely survived them, whereas a bullet can make a fairly targeted impact on the cortex or even a subcortical area. It's unfortunate that this was one of the gateways into studying localization of function. Another very famous angle on this was Phineas Gage. Here's a picture of him. He suffered an accident to his cortex. He was blasting rock, and his tamping iron, which he's holding in the picture on the left, went straight through his head and destroyed a huge chunk of his left frontal lobe. So his personality was reportedly changed quite a bit, although there's a lot of debate about how much change there was. This again stimulated a lot of curiosity about how localized different functions are. And sort of one false start was phrenology. And then from there, there was a lot of curiosity that could be partially satisfied by looking at animals. And when I was once sort of tracing the history back, I found this is probably the earliest example of a lesion study in animals. In 1888, Brown and Schaefer looked at removal of the entire temporal lobe in several monkeys. It's funny to read this paper because of this archaic terminology. So one of the monkeys, they described it as having a peculiar idiotic condition. She's tame, exhibits no fear of mankind, but shows uncontrollable passion on the approach of other monkeys. So now here's some diagrams showing this very, very large chunks of the brain that were removed. So this was the first time that the temporal lobe in general, which in humans is this shown in green here, was associated with emotion. So eventually the localization people were able to zoom in a little closer and they found that the amygdala was particularly important. And here I'll link emotion with motivation because of the types of things as we'll see. So lesions to the amygdala, the hippocampus, and adjacent areas, similar but different. And lesions to the amygdala in particular could cause, in monkeys, Kluver-Bucy syndrome. This was discovered in the 1930s. And symptoms included being very docile, calm, overeating or inappropriate eating, putting odd things in one's mouth. There was heightened or altered sexual drive. There was inability to recognize familiar objects. And in humans, you don't see exactly the same set of symptoms, but some overlap. So by the 20th century, we're starting to zoom in and identify how the amygdala can relate to emotion, motivation, and personality. And of course, as I said, emotional learning is important. And again, we kind of have a simplified understanding of what that means. There's the famous Pavlovian or classical conditioning, where the animal doesn't really have to do anything. It's just that an unconditioned stimulus, which the animal instinctually or naturally responds to, like the smell of food or the sight of food, will cause salivation. And you pair that with a bell, and eventually the bell on its own will produce the same response. Perhaps more important for the development of psychology and neuroscience was instrumental or operant conditioning, made famous by B.F. Skinner, one of the leading lights in behaviorism. Loosely, we can say that it's synonymous with reinforcement learning, which is what the machine learning community has exploited quite a bit lately. So there's the classic Skinner box, where you have an animal that has to perform some instrumental action, like just pressing a lever, and only then does it get a reward. So in this case, the active participation of the animal is required. And even though these are very simple, they are strongly associated with emotion, because they're about sort of just the basic appetitive, aversive, pleasure and pain. And so again, the brain areas that we'll talk about have often been associated with these phenomena. So starting with these basic procedures, a vast literature has accumulated, but the basic story is that the empirical study of emotion involves human studies of damage and correlation, animal studies where you actively manipulate something in the brain. And we also look at more basic connections. So the anatomy can tell us how different brain areas are connected to each other and what they look like. But that cannot really tell us anything about function unless we start by knowing something about function to begin with. From the correlations. And then there's physiology. We can stick an electrode in or some other measure like BOLD, fMRI, and see how the brain responds to emotional situations. So soon we'll see what computational perspectives can add to what's being collected here. So here's just a diagram of some of the key areas. Each of these you could sort of double click on and look at a vast collection of data, but they summarize into something that's been called the limbic system. Limbus in Latin means a boundary or border, and it referred originally to this singular gyrus here, which was kind of a border territory in the brain on the medial surface. And these two circuits, the Pepys circuit and the larger limbic system, are strongly associated with emotion and motivation and interoception. So we'll touch on some of these areas soon. So once you have a group of areas, it's sort of like collecting puzzle pieces. We know such and such areas involved, but that doesn't really tell us the larger picture. What are these things doing together? What's the connection? So it's like a puzzle. So we need to identify how the puzzle pieces fit together, which in the case of the brain is how they interact with each other. And that's the circuit perspective. Here also we have a nice bird's eye perspective that's accumulated over the years. There's something which we often call the perception action loop. We have receptors that monitor or receive signals from the environment. There's processing of various sorts, and then there's interaction with the output, the attention and voluntary movement. So this is a classic loop which is often associated with the cortex because there's so much. The cortex is sitting right on top of the brain on the surface, and it's easy to study, and there's so much of it. So you might have some further functional classification. For instance, interposed between stimuli and responses are plans or programs. So this is something to pay attention to. What are they? And do they really only involve cortical areas? Where do these plans come from? And here's where some of the areas we talked about in the limbic system will come in. So this is an extreme simplification. And as I said before, all diagrams are models, and therefore they're going to miss out something. I have ignored many key features that are of the subcortical structure. But the idea here that we were trying to capture is that there's a sensory hierarchy from sort of fine details to a coarse, rough sketch of what's out there. And similarly, there's a kind of coarse, rough sketch of what you would like to do, kind of a rough plan, and more detailed execution. In order to construct a plan, you need some kind of assessment of what the body needs. And one area that seems to be involved in this is the amygdala in conjunction with several other areas. Here I will get into this connection with the thalamus, a key source of input to cortical areas, in just a moment. So just to complete this idea, plans we sometimes think of them as highly cognitive, when you're planning something in chess. But in an ecological situation, there's families of activities that have to do ultimately with survival. And based on what you need, based on what the body needs, your planning systems need to respond accordingly. So if your blood sugar is low, for instance, you may need to eat. And so if your temperature is going up, you may need to cool off somehow. So there's several areas involved in this, and as you can tell, they overlap with the limbic system. So it's a big rabbit hole there, but there's lots and lots of data on how these medial areas, both cortical and subcortical, are involved in interoception, which is the sensing of the state of the body. And this is crucial to determining what the body needs and linking it to what is sometimes known as an affordance in the environment. Which of the things in the environment can actually help me achieve one of several goals I might have based on what I need. Right. So diving into a specific connection that this circuit turns up, there's something that our lab, the Barbas lab at BU, uncovered. So this is a reconstruction of the monkey brain. And the amygdala sends projections to something called the thalamic reticular nucleus. It's an inhibitory region that sends projections to the main thalamus, the dorsal thalamus. So one goal was, what does this connection do? That was one of my modeling goals in the broader context of emotion and attention. But why attention? How did that come up? So the connectivity of the TRN is quite distinctive. It's interposed between cortex and thalamus. It only sends inhibitory projections to the thalamus, and it receives excited projections from cortex as well as thalamus. And thalamus and cortex are well known to mutually excite each other, forming a thalamocortical loop. Now, just looking at this connectivity, Francis Crick, the same Francis Crick who was instrumental in solving the DNA structure, he became interested in consciousness, and he saw this and thought it could be an attentional searchlight. His reasoning was fairly straightforward. It was the idea that because the thalamus is the main source of input to cortical areas, other than cortical-cortical communication, the TRN seems to act like a gate, allowing sort of the transmission of sensory information as well as higher order thalamic information. So it's a good, from an anatomical perspective, potentially powerful. And now there's a lot of data showing that attention as well as what we might call pre-attentive processes do seem to involve the TRN. It's also involved in sleep and arousal. So it's not the classic attentional circuit. You'll often hear the parietal cortex is involved in that, or frontoparietal interaction. So it may be something more basic or broad than the traditional attentional circuit. And to redraw that circuit from a slightly more modeler's perspective, we can say that this is a recurring motif. So every cortical area has its corresponding TRN area and thalamic region, at least one thalamic region. And if you connect it like this, you see that if a modeler looks at this, they'll say, aha, this looks like a circuit that can implement competition. So what that means is that you have these parallel corticothalamic loops capable of inhibiting each other using the thalamic inhibitory connectivity. So this is a powerful mechanism that's quite well understood, and many modelers have used this to selectively amplify one cortico-thalamic loop representing some piece of information or a task or a plan of some sort, and inhibit something that's distracting or something that you don't want to do. And we will use this motif in the model. So as I said, the TRN can be divided into sectors depending on corresponding thalamic regions. So there's a prefrontal or frontal region, and then the sensory regions. And the amygdala, interestingly, it was found in 2012 that the amygdala projects to the TRN. It was an unknown projection before this. And not only does the amygdala project to frontal areas, as you might expect, based on its strong connection with other frontal and medial prefrontal areas particularly, but it also kind of colors outside the line, which is relatively rare. Another area that does this is the posterior orbitofrontal cortex, which is another emotion and interoception related region. The idea here is that emotion-related or motivation-related areas can have a very broad and powerful impact on the thalamic reticular nucleus, and therefore on signaling from the thalamus to the cortex. So the question is, what can this do? We have this powerful connection. What is it capable of? But stepping back again to the broad perspective, where do we start? This is, again, something that students who start in a computational program will often wonder. They often dive into large amounts of behavioral data, neural data, and then their advisor wants them to start building a model. But like, now what? Now, when in grad school, we had a professor, Ennio Mengola, who would end every lecture on the visual system with the question, what are the units? I used to find it somewhat baffling, this question initially, but now I realize it is perhaps the most important question a neuroscientist can ask, especially if they're interested in a theory or model. And by unit, I mean all sorts of things, and all of them are relevant. So there's the units of measurement. There's millivolts, for instance, so milliseconds, if you're looking at a spiking phenomenon. There's, it could be much broader units. You may be looking at something quite coarse-grained. So those are slightly different units. Or it could be completely different. Bold, blood oxygen level dependent signal is not an electrical signal per se. So these are units, and they tell you about the kinds of phenomena that you're trying to model, and they constrain what kind of model you'd like to build. There are also behavioral units. So these are plots over time of the response of an animal in different behavioral paradigms. If you are trying to model something like this, you have to keep that in mind and frame your model in terms of them. And you may have, if it's a psychometric phenomenon, so like a discrimination between stimuli, there will be units of this sort. So that's sort of on the physiological and behavioral side. But this question of units is also a structural and anatomical question. If you're interested in sort of very microscopic phenomena, such as how spines work, how synapses work, you may need a software like Neuron or Genesis, or you may even have to make your own software. And this can be very time-consuming simulations. So if you were to study the entire brain at the level of individual synapses, your simulation would run forever. So you need to make choices about what you'd like to simulate. You may coarse grain from something very detailed to something simple, like an EI model, if you're interested in, for instance, gamma oscillations. So E would be excitatory and I inhibitory. And this is a simplification compared to a detailed synaptic model, but it can capture many things of interest. And you can still go higher up and say, well, I'm not just interested in these little mesoscale circuits. I'm interested in something broad. Perhaps I want to explain the overall pattern of activation in fMRI. And there's even sort of collective phenomena. This is a flocking behavior in birds. So you may actually want to abstract the entire organism's behavior down to a point because you're interested in collective activity. So these are pragmatic considerations, because you can't model something at the level of individual synapses and expect to get your simulation to work for the broad social level phenomenon. So there's quite a challenge. So considerations of how long you'd like your simulation to run and which phenomenon you're really interested in have to be taken into consideration. So I've done all that, and you can look at the paper to see how we chose this for our particular phenomenon. But another aspect is that a model typically can't model everything, cover everything that's in a behavioral paradigm. There's often pre-training, and there's many aspects of it that are quite challenging to frame in a model. So you come up with a simplification of what you've observed in the behavior, in this case, something that's probing motivation. So in this case, let's set up a toy problem. So imagine that we have an animal like a rat that has two broad goals. There's two kinds of stimuli in the environment, or potentially. There's stimuli associated with shock, so say predators, something negative or aversive, and a food reward. So there's avoiding dangers and pursuing food. The rat must learn which sounds predict food and which predict shock or cat. So what the animal is doing in a rich environment, which might have mixes of both kinds of stimuli, is decide between two broad behavioral plans. Should I be fearful? Rodents will often freeze if they don't have a good place to hide, or should they hunt around for food? And this can be a complicated cost-benefit analysis. So a classic way that people study this, and there's lots of data on this, which associate amygdala as well as other brain regions that we identified, is pairing a neutral stimulus with new consequences. So some sound comes on, and eventually through exploration of the consequences, the animal realizes, that's the dinner bell in the kitchen. Or there could be a sound that is a prediction that the cat is on the way. Initially, these sounds had no meaning. So they were not instinctually learned. They had to be acquired. The associations had to be acquired. There's another thing that influences behavior is extrinsic biases. So it's not simply the case that one assesses what's in the environment and acts accordingly. There's, like we said, the interoception plays a big role, and there's other ways that you could bias. So for instance, this additional bias might make you much more likely to forage. So on the one hand, there may be additional information that earlier there was one cat, now there's two cats in the house, or there may be a particularly high level of hunger. So you may modulate your prior experience accordingly. You might want to be more risk-taking or less risk-taking. So our model, the Emotional Gatekeeper, it takes this basic toy problem and maps it onto a simplification of the circuit. We have in here projections from the amygdala to the TRN of a sensory map, cortical areas that represent, for instance, the sound that we were talking about. And we also have a plan or behavioral map, which are these cortical thalamic loops involved in deciding what to do, perhaps at a very broad level. We're not talking about individual movements or muscles, but the general sort of genre of movement the animal decides to pursue. Now, so this is the broad sort of box of functions. But all these functions involve interaction between these boxes. So we've set up a circuit, and I'm going to walk through how it will work. The idea is that, let's say a sound comes on, and that percolates through the sensory system. And we assume that if the sensory cortical pattern corresponding to some stimulus is strongly active, then an animal is capable of attending to it or using it in some behavior. These same signals project to the amygdala. We know that thalamus directly projects to the lateral nucleus as well as the basal nucleus. And these are places where studies have shown that synaptic learning can cause an association. So what was neutral before can come to elicit fearful responses. We also know that the amygdala projects to these medial prefrontal areas that involve weighing what course of action. So in this particular case, a sound comes on, and let's say a food reward is paired with it, maybe slightly later, and that strengthens some weights, two sets of weights, one coming in a bottom-up way from the sensory input, and also top-down input from the plan that happens to be active. So this can count as bottom-up evidence, so that once you have learned something, that says, now there's a chance of food, so be ready to eat or search around for food. Similarly, you may have a pairing between some aversive consequence, and that will serve as evidence to the other plan, the competing plan. And we're using those cortical thalamic loops with the TRN to compete between them. So if there's a conflict, the one that has more evidence in favor of it will win. And we can also have our top-down bias. So if there's extreme hunger, you could inject some additional input to bias the competition. And in such a situation, there may not actually be any stimulus in the environment. And here, the top-down excitation from cortex back to the amygdala, which we know from the anatomy, these connections exist, can reactivate all the cells that previously were paired with food reward. So this plan's representation is vague in the sense that it doesn't know exactly only one food reward. So it excites a category, all the cells that have in the past been associated with food. And those in turn can cause a kind of highlighting. So it's saying, well, be on the lookout for these stimuli, because that shows an opportunity to eat. Similarly, if there's some strong additional bias, perhaps the animal is weak and therefore needs to be much more cautious, or like I said, there's new information that has arisen. So extrinsically for now, we introduced this bias, and now it's highlighting only the stimuli that were previously associated with the negative consequence. So this top-down projection and the fact that there was learning there helps you to highlight things that are relevant to the plan and suppress anything that is irrelevant. So the inhibition, both at the plan level and at the sensory level, allow you to stick to what's relevant and that is consistent with the plan that you've chosen or that you've biased. So with this broad perspective, we can sort of zoom in and look at some of the simulations. How am I doing on time, Heiko? How much time has it been?
40 minutes, something now.
Okay, so yes, okay. I think we won't take too long, but since it's a computational model, I thought we should walk through a simulation. Now, there's a lot of information on this in this next diagram, but I will try to go through it slowly. Don't worry if you don't get all of what's in here. There's a lot here. So I will try and walk you through this from the bottom to the top. What I have is time on the x-axis, and I'm looking at various pieces of information, such as the stimulus. That's what's shown here. There's 10 different possible stimuli that I'm showing. You can think of them as little sounds, and this corresponds to, say, the frequency. And then I have a reinforcement. So the blue is the positive reinforcement, red is the negative or reversive. And I've divided the time into different epochs. So the model, this sort of robot rat, if you like, is going through different types of experience. First, a couple of stimuli, these upper two here, are being associated with food. So shortly after that sound comes on, animal is rewarded, and the corresponding top-down and bottom-up weights are modified. So as you can see here, because the weight was initially low in the simulation, it wasn't particularly active. So if you look up here, I have corresponding representations in the sensory map, which is the amygdala, and in the cortex, and the salience map, which is the amygdala. So it's a sort of place code, meaning where in that region is active corresponds to the stimulus. And so when a particular stimulus is paired with a reward, the weight goes up, and eventually the amygdala becomes strongly activated. And that activation triggers the feeding plan, the corresponding plan in the plan map. Similarly, for the opposite contingency, I've paired a different stimulus. And as you can see, this gradually, it goes from yellow to dark red, which is showing that a weight is increasing. So those cells can become strongly active and trigger the opposite plan. So this first half is a training period. So it's forming associations. Then what I do is look at testing. And I present not only the old stimuli, but also distractors, which are other completely irrelevant sounds which haven't been paired with anything. But I'm not giving any rewards or punishments. I'm seeing how it responds to the existing learning. So you have to look at it a little, you have to peer at it a little, but what's happening here is that the system is rapidly shifting among stimuli, but only the ones that are salient, which have been paired with some important consequence, either good or bad. And similarly, the plan is switching between fear and foraging quite rapidly. So it's tracking the information that's available in the world. That's what this up here is showing. I can change this slightly by changing how quickly it switches from one plan to another. So in this particular situation, during testing, there's evidence that food is available, so it stays in that food plan for long. And when it's sort of looking for food, it actually ignores the dangerous stimulus. So that lateral inhibition via TRN is causing you to block something, even though it's salient. So here we have a slow switching from one plan to the other. And there's a mechanism at work here for that we can talk about. Similarly, we talked about top-down. So what does that look like in the simulation? So let's say that there's additional strong hunger. So in this particular case, as we saw, these two stimuli corresponding here were paired with food. So when there's strong hunger, for instance, which is shown here by additional activation, keeping the plan, food feeding plan active, what you see is that those stimuli do pass through system. So the system is capable of attending to these stimuli, but the other stimulus, which was paired with the punishment, is ignored completely. So even though it's present, as you can see, well, along with distractors, so it's treated as if it was a distractor and completely ignored. So this is a situation where even though something is dangerous, it's being ignored because the drive to eat is overcoming everything else. Now, that fast and slow phenomenon is a form of behavioral switching. And we put that in there to deal with a phenomenon I'll get to in just a sec. So as I showed you, the first plot had this, and then you had a slower version where it was switching not quite on every stimulus presentation. So when you have a circuit like this that has positive feedback, we have excitation going back to excitation. One danger is that you can get stuck in a positive feedback loop from which it is hard to escape. And this is a very common phenomenon in systems that do have positive feedback, and which appears to be widespread in the brain. So it's something that needs to be solved. We don't necessarily understand how the brain solves it, but there's various mechanisms all that seem to help. So what we introduced here is that we used local interneurons. We know they exist. We don't necessarily know that this is what they do. So it's sort of a prediction of the model that local interneurons are using expectation confirmation as well as expectation violation to decide when to reset. So think of it like this. If I got some wind that there's food in the environment, I might think, oh, maybe I should wait around and start eat and look for food. But after a while, if nothing is forthcoming or if the plan is failing, I should probably reset and move on to something else. So the timescale at which this reset happens can be changed. And that's how you can switch from fairly rapid reset, which is that you're very impatient, for instance, and if you don't get what you expect immediately, you just move on to the next thing, to something maybe more patient, that will wait. I don't necessarily expect it to happen immediately, so the interneuron can change that. So this didn't emerge when I was initially building the model, but as a consequence of this feedback, I realized I needed something like this. So it came out of low-level considerations in the model that can then be interpreted in a functional way. And this happens a lot, and it's something I think modelers should talk about more because it's distinctive to modeling, but it's something worth talking about with experimentalists. Sometimes there's some need intrinsic to just the computational model that enables one to revisit the experimental literature with a new perspective and say, I needed to put this in there purely to get the model to work. But now when I look back at the literature, I now see that some of these results make sense from this perspective. This is one way in which you can get this loop between experiment and theory that can be quite productive. Obviously, you need to be careful about reading too much into the data, but theorists really should stick their neck out and try things out. So this resetting can actually go wrong. So what happens if you have no ability to reset? You have an extreme situation of behavioral inflexibility. So even though nothing resembling your expectation shows up in the world, you just get stuck because nothing seems to bring it back down. Neither expectation confirmation nor violation seem to have any effect. And this we propose is an abnormal mode. Similarly, I'm not going to show it, but you can get a lower level phenomenon where you don't get switching among stimuli. So you keep a particular, you may be able to switch between plans, but once you've expect a particular stimulus to be active, you kind of perseverate on that particular stimulus. And all of this involves local interneurons. So yes, that's what I think. I do show it here actually. Yes, but maybe we can talk about that later. So for this paper, we also did a spiking version. We were not initially doing this, and the reviewer said that it would be more impressive if the neurons were spiking. And this comes back to this question of what are the units. We initially thought that because we were looking at a circuit level phenomenon, a rate-based model that is in terms of just the firing rates would suffice. But I happened just for my own curiosity, I started doing the spiking version on my own. So my collaborators and the reviewer was very happy to see this, although qualitatively it shows the exact same phenomenon. I think this is a good thing. Sometimes people say that if a model doesn't have spikes, it's not good enough. But I think this is overly focused on potentially irrelevant details. Sometimes spikes matter, and there may be phenomena for which you do need the spikes. But we will not make any headway in creating generalizations and simplifications if every little detail of every vesicle or every molecule matters. So it's good to know that there are phenomena at the network level that are quite insensitive or invariant or robust with respect to little details of implementation. So we also tested something called emotion-induced blindness using this paradigm, where if you have something that was previously associated with, let's say, a negative stimulus, and you soon after that show a neutral stimulus, the subject will often simply not see it. And our circuit using this inhibitory lateral inhibition from TRN was well suited to recovering this sort of behavior for the same reason. Once there's an association, you get this top-down plan, and that kind of acts to block things that are irrelevant to that plan. And it depends on how closely one stimulus is to the next. If the system has enough time to reset, then you don't see this effect. And that's what this plot is showing. Let me move on. So our EmGate emotional gatekeeper model, we interpret it with an eye on psychiatric disorders. So post-traumatic stress disorder, for instance, and phobias are accompanied by altered attention. People might be skittish or suddenly turning and looking at things that other people might not bother about. And so these symptoms could arise through strong top-down biases. We model them extrinsically, but a more complicated model could explain how those arise too, these biases. And also there are low-level mechanisms that could also go wrong potentially for genetic reasons or some sort of other effect, where such as the switching, the interneurons could be damaged in some way, and that could damage flexibility. So there are multiple mechanisms potentially that could produce similar abnormalities of attention. So just to wrap up, I'd say that the forward models I talked about can allow us to do quite a few things beyond just fitting data and summarizing what we already know. We have full control over an artificial or in silico system, so we can perform virtual experiments. they're not yet, that may not be feasible in lab animals or humans. For instance, like what we did, we lesioned a specific interneuron class and looked at what happened. Optogenetics is starting to approach this, but it's still early days. And we can show how just the same circuit can perform different types of processing depending on input. Very often I see people sort of drawing two different circuits to explain two phenomena involving the same brain region. And this, from a modeling perspective, is somewhat frustrating. You really want to use the same circuit to show multiple effects. And hopefully entice experimentalists to think of new hypotheses. It might not be exactly what we simulated, but with good interaction, people may be able to think of new experiments to perform. And once again, as a reminder, all models are wrong, but I think that neuroscience may not have fully tapped into the potential usefulness of models, even if just to clarify and test our intuitions about mechanisms. And with that, I'd like to thank you for having me here. I must thank my collaborators, Helen Barbas, Dan Bullock, and Basilis Zikopoulos, and the funding agencies. So happy to take questions. I hope it wasn't too long. Happy to take questions now.
Yeah, thank you, Yohan, just for this dive through the different elements of just building models and just taking care of all the data and then just coming up with hypotheses even. I have to actually stop first the recording so that everybody might then afterwards feel, oops, where is my mouse then? Oops. Stop it, right?